Welcome back, guys. Now, in this video, let's begin from the general physiology. So, in general physiology, the first topic that I'm going to take up is cell membrane. Okay, simple topic but important for the exams. You can definitely expect one MCQ that will be coming in your exams, either a NEET PG exam or FMGE exam, doesn't matter. One question is going to be definitely there. So, now everyone knows that our cells are having a membrane around them. Okay, every cell is having a membrane, so that's called as a cell membrane. And not only the cells, even the cell organelles, they contains this membrane. So now the question is, so this cell membrane is made up of what? It is made up of carbohydrates, proteins, as well as lipids. Okay, now you can see here the cell membrane, it is having, it's a phospholipid bilayer. Even though I am calling it as a phospholipid bilayer, don't think our cell membranes are mainly made up of lipids. Okay, so we are saying like the cell membranes are made up of the phospholipid bilayer, sir. So usually what a student thinks is a cell membrane is mainly made up of lipids. No, actually 55% of the cell membrane, okay, 55%, actually there is a range 50 to 55. So 50 to 55% of my cell membrane or your cell membranes, they are made up of proteins, guys. Okay, they are made up of proteins. Then what about the lipids? Lipids somewhere around 45%. Okay, somewhere around 45. Okay, so proteins how much? 55%. Lipids how much? 45%. So then what about the carbohydrates? Yeah, even in the cell membrane there are some carbohydrate groups are also present. Carbohydrates are somewhere between 1 to 3%. Okay, actually there are ranges. So here I am giving the round figure values, better values, best values. Okay, so now tell me, our cell membranes, they are made up of carbohydrates, proteins and lipids, and what is the maximum composition? The maximum composition is always being the proteins. So this is the maximum composition of the cell membrane. Now, what is the protein to lipid ratio? In your exams, if they ask you, what is the ratio of the proteins to lipids in the cell membrane? Then your answer should be 1:1. Okay, this is the best answer. Okay, if you take the proteins out of the cell membrane and lipids out of the cell membrane and if you weigh them, so they will be in 1:1 ratio. Next, see, just now I have told you how much proteins are there in the cell membrane. 55% of the cell membrane is made up of the proteins. But there are certain membranes where the proteins are even greater than 55%. Okay, even greater than 55%. So what is that membrane where the proteins are more than 55%? So which membrane, sir? The answer will be inner mitochondrial membrane. Inner mitochondrial membrane. Mitochondria, they are having the outer membrane, inner membrane, and in the inner membrane of the mitochondria there are different complexes which are helping in the oxidative phosphorylation, ATP production. So in the inner membrane of the mitochondria there are different complexes present. And what are those complexes? Those complexes are nothing but proteins. No, so those complexes are nothing but proteins. So in the inner membrane of the mitochondria there are even more proteins present. More than 55% of the proteins are present in the inner membrane of the mitochondria. And one more best answer is presynaptic membrane. Okay, presynaptic membrane. So there is something called as a presynaptic membrane, postsynaptic membrane. In between them there is a synapse. So from the presynaptic membrane, neurotransmitters will be released. So presynaptic membrane is also a place where there is a lot of proteins. Okay, next, now let's discuss about some exceptions. What is the exception, sir? We know the cell membrane is mainly made up of proteins, but there is a membrane where lipids are greater than proteins. Okay, where lipid composition is greater than protein composition. So what is that membrane, sir? See, here in this membrane 80% lipids are there and proteins are only about 20%. Okay, 80:20. So what is this membrane? The answer is Schwann cell membrane. Schwann cell membrane, that is nothing but the myelin. Okay, so Schwann cell membrane. Now students will start to have a doubt. Sir, what exactly is this Schwann cell? If you are especially a first year student, you will have a doubt. Sir, what exactly is this Schwann cell? See, the Schwann cells, they will form myelination. They will form what? Myelination. Around neurons. in the peripheral nervous system. Okay, so who forms the myelination around the neurons in the peripheral nervous system? The answer is Schwann cells. Then what about the central nervous system? So in the central nervous system, the myelination is formed by, okay, here itself I am telling you, myelination around neurons in the central nervous system is formed by a cell called as oligodendrocytes. Oligodendrocytes. Oligodendrocytes are oligodendroglial cells, you can say. So these oligodendrocytes, what do they form? They also form the myelination. They also form the myelination, but around the neurons in the central nervous system. But Schwann cells, they form the myelination around the neurons in the peripheral nervous system. I hope you know what is the difference between the central nervous system and peripheral nervous system. Central nervous system means the brain and the spinal cord. Peripheral nervous system means the cranial nerves and the spinal nerves. Now, let's go further. Now, let's see what are the different types of proteins which are present in our membranes. See, if you look here this is the membrane, our cell membrane. Okay. Now, in this membrane, see, there are three different types of proteins present. See, here is a protein number 1, protein number 2 and protein number 3. There are three types of proteins, 1, 2, 3. So, what are these three different types of proteins, sir? Look at the first type of protein. See, this protein is extending from the outside till to the inside of the cell. From the extracellular side to the intracellular side, this protein is extending. Okay, so it's present throughout the membrane or passing through the membrane. So such type of proteins are also are called as the integral proteins or transmembrane proteins. So what is the first type of protein called as? Integral protein, also called as a transmembrane, because it is passing through the membrane from outside to inside. The second type of protein, look, the second type of protein, it is attaching only to one side of the membrane. So either it can attach towards the inside or either it can attach towards the outside, either in or either out. Okay, so such type of proteins are called as the peripheral proteins. So first one is called as the integral protein and second one is called as a peripheral protein. Look at the third type of protein, the proteins where they are attached with some lipids. Okay, they are attaching with some lipids in the cell membrane. So the third type of protein are called as a lipid anchored protein. Okay, they are called as a lipid anchored protein. So tell me how many types of proteins are there? There are mainly three types of proteins. Integral proteins or transmembrane proteins, peripheral proteins and lipid anchored proteins. Three types of proteins are there. Now, why you should know this for your exams is because they will ask you the examples. Sir, can you give me any examples of the integral protein? And can you give me any examples of the peripheral proteins and lipid anchored proteins? Now look here. See, the first type of protein, integral protein or transmembrane proteins, the examples include all the channels like sodium, potassium, chloride channel. So wherever you see a sodium channel, potassium channel, chloride channel, so they are all examples of what? They are all examples of the transmembrane proteins. Why? For example, if you see here, this is a cell membrane. See, this is how the sodium channel will be. Okay, this is how the sodium channel will be. What the sodium channel will do? It will allow the movement of sodium from outside to the inside the cell, because from high concentration to the low concentration. So this sodium channel or potassium channel or chloride channel, see, they are proteins which are extending from outside to the inside. Okay, they are passing through the cell membrane. Okay, transmembranous. So that's why they are called as the integral proteins. Okay, so from today, all the channels like sodium, potassium, chloride channel, all are examples of what? They are all examples of the integral proteins. Next, I hope you have already heard about this protein called as the sodium potassium ATPase. Okay, sodium potassium ATPase. See what the sodium potassium ATPase will do. See, later I will teach you the sodium potassium ATPase is made up of what? It is made up of two subunits called as the alpha subunit, beta subunit. What the sodium potassium ATPase will do, especially the alpha subunit, it will send 3 sodium ions out of the cell. First, it will throw 3 sodium ions out of the cell in exchange for 2 potassium ions. Okay, so this sodium potassium pump, especially this alpha subunit, what it will do? First, it will catch 3 sodium ions inside the cell, it will rotate and kicks 3 sodium ions out. Now it will catch 2 potassium from outside and brings the potassium inside. So such proteins which are undergoing the shape change, see, the sodium potassium pump, what it is doing? See, it is not a channel, it's not just a channel, it is undergoing the conformational change. Okay, while transporting the substances, it is undergoing the conformational change. So these are called as a carrier proteins. Okay, so sodium potassium pump is not a channel, it's a carrier protein. Okay, don't think, sir, both these two are same. No. So it is also helping in the transport of some substances like sodium. So sodium channel. So it is also helping in the transport of like ion, sodium. But both are different. What is the difference? See, this sodium channel, it is not undergoing any shape change, but the sodium potassium pumps will undergo the shape change, especially the alpha subunit, which is called as a functional subunit. It undergoes shape change. Okay, so such sodium potassium ATPase, see, it is also extending from outside to inside. So it's also an example of a transmembranous protein. But remember, one is a channel, other one is a carrier protein. The one which undergoes the shape change is called as what? Carrier protein. Sodium potassium pump is a carrier protein. Next step, in your second year you will study this, like these serpentine receptors. Even first year also you will study this in biochemistry. Sir, there are receptors called as the serpentine receptors. See, these serpentine receptors are also called as the G protein coupled receptors. See, this is how the receptor going to be. Okay, this is how it's going to be looking like a serpentine. Serpentine means snake like. Okay, it is also called as a 7-pass receptor. 7-pass receptor. Inside it, it is attached with some G protein like G stimulatory, G inhibitory, GQ, something like that. So, now tell me, this protein which is looking like a snake, which is passing through the membrane 7 times, actually it is going through the membrane, passing through the membrane 7 times. So it is also an example of integral protein because it is there outside, it is having an extracellular domain and intracellular domain. Okay, it's extending from outside to inside. So such serpentine receptors or G protein coupled receptors or 7-pass receptors, they are all examples of integral or transmembrane proteins. So with this, successfully what we have completed, the first type of protein completed, that's the integral protein completed. Okay, again examples, what are the examples? All the channels, sodium potassium pumps, serpentine receptors or 7-pass receptors or G protein coupled receptors. Okay, next let's look at the second type of protein called as a peripheral protein. Okay, a peripheral protein. Now you will have immediate doubt, sir, can you give me any examples of peripheral proteins, sir? We know what are the examples of integral proteins, but then what are the examples of the peripheral proteins? See, the examples of peripheral proteins, 3 examples I am giving you. So these are the 3 examples of the peripheral proteins. What are they? Spectrin, ankyrin, dystrophin. Okay, spectrin, ankyrin, dystrophin. These are the 3 types of peripheral proteins which are only attached to the one side of the cell membrane. Okay, either in or either out. Now, where is the spectrin ankyrin present? So spectrin ankyrin, they are present in your RBC. Okay, they are present in your RBC membranes. They are attached to the RBC membranes and they are the ones responsible to give the biconcave shape to the RBCs. You know, okay, RBCs, they are having this biconcave shape. Okay, there is a concavity in the center. So that biconcave shape is because of spectrin and ankyrin. Now, what if time bad? A patient is having mutation of the spectrin. So definitely there is a mutation of spectrin, definitely the RBCs are not going to be in biconcave shape. So they are going to change their shape, sir. So they are going to change to the elliptical shape. So such condition is called as, that condition is called as hereditary elliptocytosis. So spectrin mutation is going to cause hereditary elliptocytosis where the RBCs are going to take up the elliptical shape. Then what about the ankyrin? So if the ankyrin is getting mutated, again RBCs are going to lose their shape. Now the RBCs are going to become spherical shape, okay, round shape. So this condition is called as hereditary spherocytosis. So hereditary spherocytosis is mainly because of what? Ankyrin mutation. It's also because of spectrin mutation. It can be because of band 1.3, different proteins are there, mutations. But the main mutation which is going to cause the hereditary spherocytosis is ankyrin mutation, not a spectrin mutation. Okay, so spectrin can be affected, but main is ankyrin. Next step, dystrophin. So where exactly is this dystrophin present? So spectrin ankyrin is present in the RBC, sir. But what about this dystrophin? Where is it present? Dystrophin, it is present in the skeletal muscles. Okay, it's present in the skeletal muscles and dystrophin is responsible for giving the strength, the tensile strength to the cell membrane of the muscles. So because of the dystrophin, the muscle cell membrane is like, tough. It's in normal shape and normal strength. Now, what are the important points that you should know regarding the dystrophin? See, this dystrophin gene, okay, it's a protein. No, dystrophin is a protein, so definitely it should have a gene coding for the dystrophin. So what is the gene? Dystrophin gene. There is a dystrophin gene, and the dystrophin gene is considered as the largest gene in the humans. Largest gene in the humans is what? Dystrophin gene. See, I'm not saying dystrophin is the largest protein. The dystrophin gene is the largest gene. And what is the largest protein in the humans? The largest protein in the humans is a titin. Okay, titin is a protein which is responsible for the muscle elasticity. Okay, which is in the shape of the spring. Okay, the titin is a spring-like protein which is present in our muscles. So titin is the largest protein. But which gene is the largest gene? Dystrophin is the largest gene. Okay, now because of some reason, if you are having deficiency of this dystrophin, okay, deficiency, then what do you think your muscles going to be? Your muscles are going to be strong or weak? Definitely weak, sir. The muscle cell membranes are going to become damaged easily. They will lose their tensile strength and the muscle cell membranes are getting repeatedly damaged. So the muscles are going to be affected. So dystrophin deficiency is going to definitely affect the muscles. There will be weak muscles. So this condition is called as This condition is called as Becker's muscular dystrophy. Okay, Becker's muscular dystrophy. Now, complete absence of the dystrophin means complete absence. Then it's going to cause more severe problem. Becker's is a milder variant. If a patient is having a complete absence of the dystrophin, then the patient is going to have even more severely affected muscles. So that is called as a Duchenne's muscular dystrophy. Duchenne's muscular dystrophy. Okay, so Becker's is a milder problem, Duchenne's is a more severe problem. Now, how the patient is going to present to your clinic? Okay, how the questions are going to come up in your exam. Let's say here is a patient. Okay, here is a boy who is suffering with a Duchenne's muscular dystrophy. Now, if I ask him to sit on the floor, okay, I'm giving a command to asking him to sit on the floor. Now he will sit. But you and me, when we sit on the floor, if someone asks us to raise, okay, stand up, we'll just simply stand like that because my thigh muscles, my leg muscles, my gluteal muscles, they are strong. So I can simply stand like that. But look at this guy who is suffering with a Duchenne's muscular dystrophy. After sitting on the floor, if I ask him to stand up, okay, I'm asking him to stand up again. So now he is taking the help of his upper limbs, okay, in order to raise his body. Okay, his thigh muscles, gluteal muscles, the muscles are weak. So the thigh muscles and leg muscles and the gluteal muscles are not enough to raise his body. So he is taking the additional support of his upper limbs to raise the body. So this sign is called as a Gower sign. So now tell me guys, a patient who is suffering with this muscular dystrophy, what do they have? They will have Gower sign. Okay, even the Gower sign can be seen in the Becker's muscular dystrophy, but it's very clearly seen, very prominent in more severe problem called as a Duchenne's. So Gower sign is seen. What is Gower sign? You know, a boy who is suffering with this muscular dystrophy is using the support of the upper limbs in order to raise his body. Next, now if you look at his calves, okay, upon doing the physical examination, if you look at his calves, look how bulky his calves are looking. Okay, see, his calves are looking very much bulky. So you might think, so there is a calf hypertrophy, as if he is doing some exercise. Okay, no, this is not a real muscle. This is not a real muscle. So actually the muscle whatever is present here, okay, the muscle whatever is present here, the muscle actually because of the repeated injuries, cell membrane damage, because of the absence of the dystrophin, the cell membrane is getting repeatedly damaged. So the muscles which are present in this area completely dead. Okay, now in the place of muscles, who is getting deposited? It's a fat getting deposited. Okay, it's a fat getting deposited. So because of the accumulation of the fat, the calf looks hypertrophied. So that's why we are going to call it as a pseudo calf hypertrophy. It's not a real calf, it's a pseudo calf hypertrophy because it's not a real muscle, it's a fat. Okay, pseudo calf hypertrophy because of the fat deposition or adipose tissue deposition. Okay, so done. So with this, what we have completed? We have completed the two important signs. Okay, upon doing the physical examination, you can find the pseudo calf hypertrophy and you can also see the Gower sign positivity. Now, what is the most common cause of death? So the most common cause of death, these people, they are having muscle weakness. No? So the most common cause of death is cardiomyopathy. It's a recent advance. Okay, so the most common cause of death is cardiomyopathy. Okay, heart failure. If it is not there, in your book, then you can go with the respiratory failure. Okay, respiratory failure. So previously, respiratory failure used to be the most common cause of death because skeletal muscle, the diaphragm is weak. So whenever the diaphragm is getting weak and whenever the diaphragm is getting damaged, so respiration will be affected. So respiratory failure is going to be the most common cause of death. Okay, previously, but these days because of the advancements in the respiratory medicine, okay, ventilators are available. So the most common cause of death now shifted to cardiomyopathy, heart related problem, heart muscle related problems. So if you are having cardiomyopathy in your options, go with the cardiomyopathy. Okay, so now with this, what we have completed? We have completed peripheral protein. What again? Repeat, guys. What are the three important peripheral proteins that I have taught you? Spectrin, ankyrin, dystrophin. Okay. Next, let's see the third type of proteins in the cell membrane called as the lipid anchored proteins. Okay, lipid anchored proteins. Now, what is the example of this lipid anchored protein? The one example of the lipid anchored protein is GPI. GPI. What is the full form of GPI? Glycosyl phosphatidyl inositol. Okay, glycosyl phosphatidyl inositol. Now, where is it present and what is its importance? Let me tell you. Guys, look at here. Here is a healthy RBC. Let's say here is a healthy blood cell RBC. This is not only true with the RBC, but this is also true with WBC and even platelets. Okay, platelets also. So now I'm going to only talk about the RBCs, but this is also true with the RBC as well as the platelets, that you have to remember. On all the blood cells, this GPI protein is present. You can see here there is a lipid anchored protein called as a GPI. It is attached with some lipids. Okay, it is tightly attached with some lipids in the cell membrane. Okay, sir, GPI. Now what this GPI is doing? You can very clearly see this GPI, it is holding a one more molecule here and here. So what is this molecule? What is the name of this molecule? CD55. And what is the name of this molecule? CD59. So this GPI, it is holding two molecules called as CD55 and CD59. So the CD55 is also called as a DAF, decay accelerating factor, and CD59 is also called as a MIRL, membrane inhibitor of reactive lysis. Now you will start to have it out, sir. What exactly is this DAF and what exactly is this MIRL? What they are doing? Let me tell you. Guys, listen. Imagine you are having an infection. Okay, imagine you are having some infection, sir, because of some bacteria. You are having some infection today. Now, definitely tell me, is it good or bad? Definitely it's bad, sir. Now, whenever you are having some bacterial infection or fungal infection, so this bacteria is going to activate something called as a complement proteins in our blood. Complement proteins. You know, it's a part of the innate immunity, complement proteins. So, complements, now once they are activated, this activated complements, you know, there is something called as a classical pathway, there is something called as an alternate pathway. Okay, so because of this complement activation, complements, they are going to form membrane attack complex. Okay, what is forming? Membrane attack complex is produced. At the end of the day, membrane attack complex is produced. Now this membrane attack complex, okay, which is nothing but the activated complements. What they will do, see in the name itself, it's their membrane attack. They will attack the membranes. Which membranes? Now this membrane attack complex, it will come and attack the bacterial cell membranes. Okay, it will come. What it is doing? It is going to attack. Okay, it's going to attack the bacterial cell membrane, and it's going to put, actually it's going to drill, it's going to put the drills, okay, pores in the bacterial cell membrane, and it's going to cause the death, lysis of the bacterial cells. And that's actually good. That's actually good. We want to kill the bacteria at the end of the day. Okay, fine. But the problem is this membrane attack complex, okay, it's like a blind soldier with a powerful weapon. Okay, it's like a blind soldier with a powerful AK-47. Now what I'm trying to say, now when this membrane attack complex is produced, it's going to, its main goal is to attack the membranes. So it's not only going to attack the bacterial membranes, but it will also start to attack our own cell membranes. Okay, I have told you, it's like a blind soldier with the AK-47. So this blind soldier doesn't know who is enemy and who is our own cell. So now this membrane attack complex, what it will try to do, it will try to attack our own cells also, like RBC, WBC, platelets. So tell me, there is a chance that hemolysis can occur. Tell me, is it good or bad? Definitely bad. We should not kill our own cells. So our cells are having their own protection. Our cells are having their own protection. What is that? So on our cells, there is something called as a DAF and MIRL, CD55 and CD59. So whenever the activated complements, whenever they are trying to come and attack our cells, whenever they are trying to come and sit on our cells, now immediately this DAF and MIRL, what they will do? They will inhibit this activated complements. They will destroy this activated complements, so that the activated complements will not kill our cells. So our RBC, WBC, platelets, they are having their own protection in the form of DAF and MIRL. And who is keeping DAF and MIRL on the surface of the cells? It is GPI. Okay, so now you think, what if today I am losing this GPI? The GPI is absent on my blood cells. Now on my RBC, WBC, platelets, now GPI is not present. So now do you think DAF and MIRL, the CD55 and CD59, do you think they can sit on the cell membranes? No, definitely not. So now what will happen? Now the membrane attack complex, now it can attack our own RBC, WBC and platelets. So what will happen? So now my RBCs are getting damaged within the circulation. Now my RBCs are getting damaged. So that is leading to hemolysis. Okay, hemolysis, RBC destruction, platelet destruction, even WBC destruction. RBC destruction will lead to what? Hemolysis. WBC destruction will lead to what? Infections. Platelet destruction will lead to what? Thrombosis. Okay, so now this condition is called as, okay, paroxysmal nocturnal hemoglobinuria. Okay, we'll discuss one by one. We'll go one by one. See, this GPI, what this GPI is doing, it is trying to prevent, it's preventing the damage of our blood cells from the activated component. GPI is good. It is trying to prevent the RBC destruction, platelet destruction, and WBC destruction. What if time bad, if this GPI is absent? If GPA is absent, that will lead to a disease called as a paroxysmal nocturnal hemoglobinuria, and it is the acquired hemolytic anemia. It's not inherited disorder, it's acquired disorder. So I am going to acquire, because of some mutation, I am going to lose this GPA. So it's acquired hemolysis is happening, hemolytic anemia. Now this is because of which gene mutation? PIGA gene mutation. Okay, if my PIGA gene, if it is mutated, I will lose my GPI. So if my GPI is not there, if there is no GPI right here, if there is no GPI, there is no CD55 and CD59 on the cells. So what will happen? The membrane attack complex. The membrane attack complex is going to attack my RBCs. So that's why hemolysis, that's why there is hemoglobinuria. RBC is getting destroyed, so hemoglobin is going to leak into the blood, hemoglobin will filter into the kidneys and leading to hemoglobinuria. So the person, whenever he goes to the urine will be dark in color because of the presence of hemoglobinuria. Okay, now what is the most common cause of death? If you ask me, the most common cause of death is a thrombosis. Okay, why thrombosis? I have told you, it's not only the RBC destruction but also platelet destruction. Platelet destruction is going to release certain products which will activate the thrombosis, intravascular clotting. Okay, disseminated intravascular clotting. Okay, so thrombosis, especially in the in unusual places. Okay, like in the hepatic portal veins. Okay, so which is called as a Budd-Chiari syndrome. If there is a hepatic portal vein thrombosis, that's called as a Budd-Chiari syndrome. So yes, thrombosis is going to be because of the platelet destruction. Is the most common cause of death. Why infections? Because of the WBC destruction. Whenever you are losing the WBC, you are compromising the immune system. So that will lead to the infections. Okay, so that's it. And if they ask you, what is the gold standard investigation? The gold standard investigation is to check whether the GPA is present on our blood cells or not. How to check whether the GPA is present on our blood cells or not? The study is called as flow cytometry. Okay, very important. A lot of times this question has been asked in the NEET PG exams. So flow cytometry, with this study called as a flow cytometry, you can actually check what are the different components which are present on the cell membrane, whether the GPA is present or not, CD55 is present or not, CD59 is present or not. Okay, so the gold standard investigation is flow cytometry. Okay, so with this, what we have completed, guys, we have successfully completed the cell membrane composition and what are the different types of proteins which are present in the cell membrane. Okay, now in the next video, okay, we'll be discussing mainly about the different types of lipids present in the cell membrane. Thank you.